Dear students, after studying this module, you shall be able to know about the significance of biological and non-biological matrices, the forensic importance of biological matrices, and the methods applied for the extraction of poisons in biological matrices. Poisoning has turned out to be one of the most commonest emergencies all over the world for the reason that thousands of pharmacological and chemical agents are frequently used and their number is going to increase every year. While studying the toxicology in general, the stress is given upon the typecast substances known having poisonous nature. The methodology of extraction is the paramount step in the qualitative and quantitative analysis of poisons from complicated biological samples. The enormous collection of substances that are toxicologically significant, such as pharmaceutical medicines, herbicides, insecticides, etc., contrast conspicuously in their physicochemical properties. They are habitually potent agents that are present only in very low concentrations. For that reason, a particular extraction procedure may not only be able to separate the target substances from interferences in the specimen, but also be able to increase their concentration comparative to the co-extracted matrix compounds. The extraction process is every so often time consuming and labor demanding, but practical participation shows that the effort is necessary because the consequent analytical steps are based on and benefit from the sample preparation. The isolation of the compounds of concern from the biological matrix is important for their efficacious detection, identification and quantification. The approach applied and the effort spent in the development of an extraction procedure depend not only on the physicochemical properties and the probable concentration of the target compounds, but also on the nature of the specimen and the available apparatus in the analytical laboratory. Occasionally, the physicochemical properties of the target compounds allow for their detection and after digestion of the sample matrix or for an easy separation from the less volatile matrix components. However, there is a significant group of less volatile drugs and poisons that demand more intricate extraction procedures to make certain of their isolation from the biological matrix. There are choice of appropriate technique of extraction which depends on a number of controlling aspects such as the nature of poison, matrix or matrices, and also upon the extent of samples available or forwarded for examination purpose. The active constituent should be extracted from sample in least possible stages so as to avoid loss in the course of handling. Now we will study about the forensic issues. Blood and selected viscera are generally collected during medico-legal examination in suspected cases of poisoning. This is sent to the forensic science laboratory or the FSL of the region, and based on the analysis that has been conducted or that has been carried out, concluding opinion as to the cause of death is then furnished based on the toxicological findings. Forensic toxicological analysis has conventionally focused on the use of blood, body fluids and certain organs in examination of death due to intoxication. However, in some circumstances, putrefaction and contamination make accurate sampling from tissues difficult, such as in cases of exhumation. In these cases, the bone marrow might be advantageous as an alternative specimen since it is a potential depot for drugs. Biological matrices or viscera containing fats, degraded protein and coloring matter, etc. makes the extraction of active constituent very challenging. Because of the large number of toxicologically relevant compounds and the different questions that need to be responded in the various fields of analytical toxicology, a universal standard extraction procedure does not exist. Optimized extraction procedures are available for many drugs and poisons. The effectiveness of extraction governs the limit of detection, accuracy and precision in the determination process. The expert plays a substantial part in the selection of suitable procedure on the basis of diverse factors like case history, quantity, physical state of matrices, analytical requirements and also the infrastructural conveniences which are accessible. Next is classification of matrices, that is, how do we classify the matrices. Generally, there are two types of matrices encountered in the toxicological analysis of the exhibits. They are the biological matrices and the non-biological matrices. So first we will focus on the biological matrices. The biological matrices comprise of viscera, blood, urine, saliva, stomach contents, intestinal contents, gastric lavage, vomit, brain matter, stool, fecal matter, bone, nails, hair and skin. Viscera consist of the internal organs of the body which are liver, kidney, stomach, intestine, gallbladder, uterus, heart, lungs, brain, etc. Next, we come on to the non-biological matrices. The non-biological matrices consist of water, remnants or traces of poison in a small container, food and food products, milk and milk product, fruits, vegetables, tea, coffee, cooked materials, drinks, cereals, pulses, wines, etc. Now students, we will study about the methodology, how to extract these. The organic matters which constitute the bulk portion are destroyed by chemical means to get the active constituents free completely for qualitative and quantitative analysis. The extraction in biological matrices may be carried out by various methods. So first here is the dry ashing method. In this method, appropriate amount of macerated tissue or other biological materials is taken in a silica crucible and heated in a Bunsen burner for eliminating the moisture and moderately rescinding the organic material. Then the crucible is kept in a muffle furnace. The temperature of the furnace is raised up to 550 degrees centigrade, and at this temperature the burning of the organic matter is made by keeping the silica crucible for about 1 hour. After the incineration is finished, the crucible is then taken out. The color of the residue is to be noted as when warm, because in presence of zinc the residue assumes a yellow color, while in presence of copper the residue will assume a slightly bluish green color. The residue in the silica bowl is boiled with 10 ml of 4 normal hydrochloric acid and then filtered off. The clear acidic solution is tested for metallic poisons such as copper, bismuth, zinc and barium, etc. by carrying out the general group analysis using semi-micro method, chromatographic and other instrumental technique. Next is the wet digestion or the acid digestion method. In this technique, adequate amount of biological material or 10 milliliters of blood is taken into a Geld-Hall flask and 20 to 40 milliliter of concentrated nitric acid is then added so as to cover the material. The flask is gradually heated in flame till the mass begins to liquefy. The heating is continued until the liquefaction of the material is complete, and that must be completed in the presence of abundant brown fumes of nitrogen dioxide in the flask. At this point, about 20 to 30 milliliter of concentrated sulfuric acid is added and the flask is heated intensely, and concentrated nitric acid is added drop by drop to the contents of the flask at the rate of about 10 drops per minute, so that the atmosphere in the flask must at no time be free from the brown fumes. Heating should be continued until all organic matter is destroyed and the liquid develops a clear, colorless or straw colored. To find out if the oxidation is complete, the flask is heated without addition of any nitric acid. If there is any unburnt organic matter, the liquid begins to darken, and if the digestion is complete, no darkening takes place and the white fumes of sulfur trioxide are released. In the previous case, the addition of nitric acid and heating are continued more till the organic matter is entirely oxidized. Heating is continued for 15 minutes more so as to dispose of the nitric acid totally. Then, after cooling, 25 milliliter of saturated ammonium oxalate solution is added. The liquid is boiled until sulfur trioxide fumes appear. This confirms the complete removal of nitric acid. It is then cooled and diluted with an equal volume of water and cautiously relocated to a beaker. The beaker is then again heated so as to dispense with the remaining sulfuric acid. The solution is then cooled and diluted with water in such a manner that the strength of the acid is about 10%. At this juncture, a precipitate may be formed which contains the insoluble salts of lead, bismuth, tin, barium, strontium or silver, etc. The precipitate is then filtered off and tested for the metals that have already been told to you. The filtrate will now contain all the other metals except mercury. It is then subjected to a systemic group analysis and quantitative determination subsequently as and when required. Next is the Fresenius and babo method. The wet digestion method of destruction of organic matter is absolutely not appropriate for mercury, which may be practically disappeared by volatilization. This Fresenius and babo method or technique is considered to be the most acceptable for deliverance of mercury, even though there is a likelihood of some loss of mercury by vaporization. A certain amount of biological material is taken in a flask fixed with a reflex condenser. In the event of viscera or any solid material, the adequate amount of water is added to make it consistent. 1/3 of its volume of chemically pure hydrochloric acid and a few grams of solid potassium chlorate are then added. The content is mixed by pulsating. The mixture is heated and small amount of potassium chlorate is added time to time and the flask is shaken. Chlorine gas evolves. The heating is continued until the contents of the flask becomes a constant straw colored liquid free from the organic matter except some fatty substances in suspension which cannot be oxidized completely. If heating for an hour after the last addition of potassium chlorate produces no darkening of the mixture, the oxidation of the organic matter or the mixture may be taken as to be finished. It takes around 4 to 6 hours to achieve this stage. It is filtered and washed with water then. The filtrate and washings are then collected again. Adequate sodium sulfite or bisulfite is then added so as to reduce the surplus chlorine into hydrochloric acid. The liquid is warmed on water bath and a current of air is passed so as to discard the excess sulfur dioxide. Next is the selective chemical treatment. In case of presence of arsenic and antimony in their higher oxidation state, that is the pentavalent state or +5 state, reduction to the trivalent or +3 state is to be carried out for the chemical treatment. A few toxic metals such as arsenic and antimony in their oxidation states may be subjected to the reduction process by nascent hydrogen for isolation of metals in matrices like burnt bones, hair, nail in the form of their volatile hydrides, that is arsenic hydride and antimony hydride. The method may also be carried out by Gutset or Marsh-Berzelius method. Fifth process is the direct solvent extraction. The biological materials, that is the viscera, are mixed with ammonium sulfate and made uniform. After addition of the diethyl ether, the mixture is shaken at pauses and set aside for overnight. It is filtered and the concentrated extract is cleaned up by passing through a chromatographic column having 3 sequential stratums of different length of 5 cm layer of alumina, that is the top layer. Next is the 2.5 cm of activated charcoal or the middle layer, and the third layer is of 2.5 cm of anhydrous sodium sulfate. This is the bottom layer, formerly washed with ether. The elute is evaporated to dryness as has been done before. Next method is the micellar extraction in biological matrices. The biological material or viscera is mixed with ammonium sulfate and then homogenized. After addition of diethyl ether, the mixture is shaken at interludes and left over for overnight and filtered off. The ethereal extract is taken into a separating funnel and sodium lauryl sulfate is added and agitated gradually. On settling, fat in liquid and semi-solid form is separated and taken off from the system. The addition of surfactant is continued till all the fatty materials and proteins are separated and settled at the bottom. The end point is specified by a change of dark color of ethereal layer to colorless layer. The ethereal extract is shaken with water twice moderately. The ethereal layer is then collected off. In case of emulsion formation, ethereal layer is collected by breaking the emulsion with excess ether and mild stirring. The collected ethereal layer is dehydrated over anhydrous sodium sulfate to remove the traces of water. The ethereal layer is decanted and evaporated to dryness as has been already told to you. Next technique is the modified Stas-Otto method. Adequate amount of biological material is pulverized in this technique and then mixed with plenty of rectified spirit in a flask and acidified with tartaric acid. The mixture is heated on a steam bath for about 1 to 2 hours with exhaustive shakings at regular pauses. The extraction is then followed to carry on for about 24 hours with the steam off. It is then filtered through a corrugated filter paper. The filter is evaporated and the residue is again extracted with acidulated alcohol in the same way as and filtered and washed several times with hot rectified spirit. The combined filtrates are evaporated in a porcelain basin on a steam bath to a viscid uniformity. To this thick viscid residue, rectified spirit is gradually added with constant stirring so that the insoluble matter may be granulated and not viscous. It is warmed with random stirring for about half an hour and filtered off. This process is repeated once more and the combined alcoholic extract are evaporated almost to dryness. The residue is now dissolved in water, acidulated with dilute sulfuric acid and filtered after about an hour. The poisons are consequently dissolved by the aqueous solution, which is relocated to a separating funnel and extracted with an appropriate solvent such as ether, chloroform, etc. The acid aqueous solution is then rendered alkaline with a solution of sodium carbonate or ammonia, which would release the free base from its salt. The alkaline solution is now extracted with chloroform in the similar manner as has been already mentioned in the various stages. It will take up all the alkaloids except for morphine and those weakly basic substances which are somewhat extracted from the acidic solution. So students, let us summarize this module. The methodology of extraction is the paramount step in qualitative and quantitative analysis of poisons from the complicated biological samples. A particular extraction procedure may not only be able to separate the target substances from interferences in the specimen, but also be able to increase their concentration comparative to co-extracted matrix compounds. Matrix or matrices are any material substance in the earth in which the active constituent may be dispersed, accumulated, left, absorbed or chemically bound. The isolation of the compound of concern from the biological matrix is important for their efficacious detection, identification and quantification. Biological matrices or viscera containing fats, degraded protein and the coloring matter etc. makes the extraction of active constituents very challenging. While talking about the biological matrices, generally blood and certain viscera are collected during medico-legal autopsies in alleged cases of poisoning and sent to the forensic science laboratory for proper and formal detection, identification and quantification. Solvent extraction is a common technique in forensic toxicology related to the biological matrices. Solvent extraction method has now been upgraded and made automatic, that is accelerated solvent extraction or the ASE technique. In cases of solid non-biological matrices, continuous extraction of a oxalate may be employed, that is the continuous extraction. Micellar extraction is a special type of extraction procedure that appears to be unique in the suppression of drugs, plant poisons and pesticides in the biological matrices.